UW360 is proudly supported by BECU, a not-for-profit member-owned credit union. Pacific Office Automation, copy, print, workflow, and IT. Problem solved.
Hi, I'm Carolyn Douglas. Welcome to UW360, and welcome to the Molecular Engineering and Sciences Building, a new addition to campus this year. We'll explore some of the cutting-edge research happening here a little later in our show. We'll also see what it takes to be an Army ROTC cadet at the UW, discover the secret weapon used by the Huskies' championship volleyball coach, and see what the UW's conservation canines are sniffing out and why. But in our first story, we're heading to Zambia. It's one of the countries where UW researchers are conducting what may be the most ambitious global health measurement ever.
Global health is an issue that touches us all. Billions of dollars are spent each year to treat illness and save lives. But does that effort and money produce results? How can we pinpoint aid to the diseases that most require attention? The Institute for Health Metrics and Evaluation, or IHME, is an independent research center at the University of Washington. They're collecting data from all over the globe to answer these questions. Their massive study is called the Global Burden of Disease, or GBD.
What we do to try to quantify all the world's health problems is we try to find all the data that we can possibly find on 291 different diseases and injuries and for 66 different risk factors. Once we get all that data, we then try to analyze it using the most appropriate statistical methods to come up with actual estimates of who dies from what, who's sick from what, and then put that all together in a comprehensive understanding of people's health problems.
Some of the results have been surprising. In Zambia, for example, it was believed that in recent years malaria mainly caused infant deaths. But the GBD study revealed that the adult mortality rate from malaria is still a problem. HIV is a known issue in Zambia, but the malnutrition data surprised the country's leaders.
Our biggest contribution to national policy debate is to actually put things in that comparative light. So to remind people that, gosh, you have much more protein, energy, malnutrition than your neighboring countries. What's going on? Why is that? And it triggers a whole series of discussions that I think would otherwise not occur if you didn't provide the internal comparison between diseases and injuries in Zambia and the comparison to neighboring countries.
Making the data they collect visual and immediately accessible to governments and the general public is another mission of the Global Burden of Disease.
So when we have the data, we have just sheets and sheets, millions of files of different numbers, and we need to find a way to come to terms with how we're looking at differences over time, over space, over age groups.
IHME makes it easier to follow data from countries like Zambia.
And the malaria epidemic, similar to many countries in the region, increases in the early 2000s and late 1990s, and then as more time and effort and resources are dedicated to global health, you can see that that starts to pull back quite a bit as we move towards 2010. And here you can see the curve over time, and we focused in on Zambia, and you can see that we really do have this hump here in the late 90s, early 2000s that we've started to combat and bring down.
The results of the global burden of disease are disseminated to decision makers around the globe.
Going forward in the future, there's a lot of things that if you also add on to this database will make the information even more useful for decision makers. We've been focused on what are people's health problems, but in parallel, if we can also track where money goes, money for medical care and public health, and then you can compare the two and say, look, we're spending a lot of money over here, but we're ignoring this problem over here. That becomes even more powerful for decision makers. So this is the period of time when it's very unlikely that somebody knows they're infected.
Nothing is more powerful than knowledge. Using independent, science-based measurement, IHME and their partners are changing how the world understands global health and how it responds. Because ultimately, every number is a human life.
To learn more about this story, you'll find a link on our website, uwtv.org/uw360. Now stay with us for a college curriculum that's definitely not for the faint of heart as UW360 continues. Welcome back, everyone. Now, when I was in college, we did everything we could to avoid those early morning classes. But one special group of very dedicated cadets is already hard at work before the sun rises.
Whose house? Our house. Hoorah!
There are key words people use when talking about careers in the U.S. Army: sacrifice, honor, strength, and adventure. And ROTC programs at colleges all over the country are designed to help prepare cadets for those careers.
I've done so many cool things I thought I would never do. I've got to fly in like Chinook helicopters. I've got to rappel down walls.
Big bound. Nice. Throw it out.
Here you get to work with people all the time. You get to help mentor people, and it's just, it's a great opportunity, and I love everything about it.
Hoorah!
Lindsay and Dallas, or Cadet Keith and Cadet Jones, are in the Army ROTC program at the University of Washington.
ROTC stands for Reserve Officers Training Corps. When a cadet serves out their final year of Army ROTC and graduates from college, they are commissioned into the U.S. Army.
The UW has ROTC programs with the U.S. Navy and Air Force, in addition to the Army program, all housed in Clark Hall on campus. Lindsay and Dallas take courses in military science to develop tactical expertise and leadership abilities. Cadets can major in anything they want. For instance, Lindsay is majoring in communication, and Dallas is still undecided.
Attention! Toward war!
But just like any Army soldier, they are required to stay in peak physical condition.
Monday through Friday, I'm up at 5:30.
They could easily choose to, hang out late at night, sleep in a little bit in the morning, but they've committed themselves to doing something a little bit different.
Don't swear about being alone!
Don't swear about being alone!
This weekend we're down at Joint Base Lewis McChord conducting our FTX field training exercise, which is geared towards confidence and team building skills for the cadets.
Cadets come to the program from all different backgrounds. Some have no experience with the military.
Are these the ones we have to get the academy?
And for others, like Lindsay, military service is a family tradition. Her father retired from the Army as a lieutenant colonel in 2008.
He was excited, but nervous like any dad, I think. I'm the youngest of my brother and sister. I'm the baby. I'm 21 years old. He wasn't surprised, I guess. I like physical activity. I like getting my hands dirty. So I think for him, he's really proud.
And the cadets are part of a proud heritage at the University of Washington.
8 medals of honor have come from ROTC graduates from University of Washington, which is second only to the military academy at West Point.
Whether coming to the program because of family tradition or a personal career choice, these cadets share a focus on their futures.
Being around people in ROTC in general, I feel like I'm a better person for it. Because I'm working really hard, physically, mentally, I'm going out and training, and I'm preparing to be an officer.
Step towards me.
And I feel like with that amount of preparation for your career, I feel like that's just, it's rewarding because you know it's going to be great, and you know that if you put in a lot of effort, you're going to get a lot out of the program.
It is fulfilling to see cadets show up in the first year, not even sure of what they're capable of doing, and then bring them along over the course of 4 years.
Whose house? Our house!
But see them develop, see them grow, show them that they can do things that they didn't think they could do, find out that they are in fact capable of doing things that I didn't think that they could do.
Crew, just to let you know, you guys now have the fastest time on the obstacle course.
Hell yeah!
I've always been really spontaneous and I just want to experience like as many things as possible in life, and through ROTC and being in the Army, I definitely know I'm going to experience a lot.
For more on the UW's Army ROTC program, you'll find a link on our website. Now, in our next story, we're going to show you just one of the incredible things happening here at the Molecular Engineering and Sciences Institute. A group of researchers is working to design ways to deliver drugs to specific parts of our bodies.
In these biotech research laboratories at the University of Washington, scientists are working to design new versions of the miniature machines that carry out nearly all the important functions of living things, proteins.
You've probably heard of DNA and genomes. Those are the blueprints, the instructions. But what they're the instructions for is making proteins, and proteins go and do all the work.
Proteins regulate the electric currents in our brains. They metabolize the food we eat. They build up our muscles. They can both cause and cure diseases. These researchers are working at the level of nanotechnology, the science of the very, very small, in order to find the solutions to big problems. One of those problems is called drug delivery.
Most drugs that you take are considered small molecules. They're really tiny and they can diffuse almost anywhere in the body. The type of drugs that we're looking to deliver are the next stage of drugs. So these are larger drugs like proteins or nucleic acids or even peptides. These are too big to freely diffuse in your body. So we're trying to design carriers that can help them get where they need to go.
Professor Pun and her team are essentially designing transportation systems inside the body.
And one of the main focuses of our lab is delivering materials to the nervous system. It's actually considered largely an undruggable target. So you'll see that a lot of the diseases like Alzheimer's or Parkinson's or injury to the brain or spinal cord, there's really no treatment for them right now. What we're trying to do is to take some of these materials and to be able to get them into the brain or spinal cord so that we can start getting drugs there and see if we can treat these diseases.
The students here work with high-tech imaging systems, identifying proteins with fluorescent markers to follow their progress.
They have fluorescently tagged our material, and they're looking at it under a fluorescent microscope. That magnifies the cell so you can see it, and the fluorophore tag allows you to track where it's going.
Professor Pun is quick to point out that a cure for some of the diseases she is trying to target is still 5, 10, 15 years or more away. But the results of this research will spur others to work even faster.
And we're just working in small animal models at this point. But what we hope is if we generate really exciting data, which we'll find out in the next year or so, that if we can get pharmaceutical industries interested, that they'll be able to pick it up and make it go much more quickly than we ever could.
So there are all these problems that the world faces, and just in the same way that biology solved the problems it faced by evolving better proteins during evolution, our hope is to be able to create new proteins that will solve some of the 21st century problems.
Engineering proteins to boldly go where no protein has gone before is not science fiction. It's real. And the work needed to make it happen is going on right now inside the Molecular Engineering and Science Building on the University of Washington campus.
Stay with us. We'll be back in a minute with the Husky volleyball team and their impressively innovative coach. Welcome back to UW360. When head volleyball coach Jim McLaughlin came to the University of Washington in 2001, he took the team from last place in the Pac-10 to a national championship in 2005. His secret weapon? A whiteboard.
It was 2011. In October, Jim McLaughlin's team was not playing to its usual high standards.
We thought, hey, we got to look at this. We need to do something different or we're not going to win a lot of matches.
McLaughlin gathers and analyzes streams of numbers. He believes carefully scrutinized data reveals much more than the eye can see.
And it was right there in front of me, and we needed to make some changes, or at least try to make some changes.
His numbers told him it was time to switch to something known in volleyball as the 6-2, a system he'd never before used at Washington.
The 6 means that you have 6 different options to set.
The setter will stay back for 3 rotations.
The 2 part of the 6-2 means there's 2 different setters.
3 hitters with the first setter and then 3 hitters with the second setter.
Now, as you might guess, it can be pretty darn tough for any team to simply change its offense in the middle of a season. Add to that ego, especially in sports. Many of us make decisions and then want to appear strong by defending those decisions no matter what.
You know, when I was young, I did it that way, and it really made sense to me.
But the more he coached, the more he was willing to learn. He realized it's not enough to merely measure athletes' performance. You have to measure the right things.
And you really got to look, you know, you hear coaches talk a lot about how they really investigate the stats and study them deeply, but you got to know what's important. You know, you have to do the cost-benefit analysis in everything you do, and you got to check and balance it, and that's what I think. You know, you got to try these things, and you got to give everybody an opportunity.
So McLaughlin switched from using just 1 setter in each match to using 2.
And we did it, and we liked it, and we were better collectively in the 6 rotations.
That's the set.
But like most changes, it caused disruption. Under the old system, for example, setters like Jenny Nogueras were asked to perform all volleyball skills, including blocking.
I like blocking. I think blocking is probably the best feeling that you could ever have is when you stuff block someone, and it's probably #1 right here, best feeling.
But running a 6-2 offense means setters no longer block. At the same time, it also means more players get to compete for time on the court.
Competition helps develop people. You know, if you put them in a competitive environment and you measure and manage things, that's really important. You know, at some point, a lot of people can serve and pass and spike, but who's going to win the really tough games? You know, who has that want to, that discipline, that courage, that fight? You know, those things. Who's going to compete the whole way through? Those things are very important, and maybe more important than ability.
It took guts and confidence to abruptly change his system. The changes worked. But McLaughlin's tinkering won't end there.
I don't know, I had a reporter ask me the other day, you know, when was your best year coaching? And this year, because I'm older, I've studied it longer, I've got to do it for a long time, and I'm going to continue to get better at it.
Earlier this year, Coach McLaughlin celebrated his 500th career victory, a very impressive accomplishment. Stay with us. We'll follow the UW's conservation canines into the wild as UW 360 continues. Welcome back, everyone. Okay, here's a job that's definitely for the dogs, UW dogs that is, who sniff out animal scat in the woods and on the ocean so researchers can bag it for environmental studies.
Tucker the Labrador has a mission.
So you can see he's got a scent.
He sniffs out scat on land and sea. It helps his partner Sam do research.
I was very interested in monitoring how the environment impacts wildlife. Many years ago, actually in the mid-1980s, I developed ways to measure stress and reproductive hormones from feces of wild baboons. And I quickly learned that there are a huge number of physiological and genetic products that are excreted in feces for wildlife, and that the feces is the most accessible animal product in nature.
For his help in finding that accessible product, Tucker gets a reward: playtime with his ball. Sam Wasser, director of the UW's Center for Conservation Biology, gets a wealth of material for his research.
There are so many biological products in there. We get DNA from the host animal, from the lining of its intestines, and that allows us to determine the species, the sex, the individual identity of the animal. From that, we can calculate the abundance of the animal. We can see how it's distributed over the landscape. We can look at sex differences in different traits. We also get stress hormones, reproductive hormones, nutrition hormones from the samples. We can get immunoglobulins from the samples, which means that we can look at the stress levels the animals are experiencing, their nutritional levels, their immune function.
This insight into the effect a changing environment has on the lives of animals is invaluable to conservationists. When habitat changes, there are always multiple factors. The data researchers collect from SCAT allows them to isolate the impact of those individual pressures.
By having this suite of physiological measures that we can get over time when various features in the environment are changing, we can use that to partition the various pressures facing these animals. That tells us what we need to fix first. So that's a really, really powerful tool that is desperately needed in the conservation and management of wildlife.
Okay, let's go find it.
It's a much more effective method than capturing and tagging animals, allowing for both more detailed data and a wider population sample. Tucker and the other dogs on the Conservation Canines team, along with their human handlers, have worked on projects around the world. They've sniffed out scat for northern spotted owls, Pacific pocket mice, giant anteaters in Brazil, tigers in Cambodia, and caribou in Alberta. Basically, anywhere animal populations may be affected by environmental change. The dogs love it.
They're hot spots. I've been doing wildlife research since 1973, so 40 years. And this is by far the most rewarding work I've ever done, because you're out there and you're working as a coordinated team with these dogs, and you just can't help but fall in love with these crazy animals because they're so good at what they want to do, and everything is all positive about play.
In Northwest waters, Tucker and his team are tracking orca whale scat. They gather samples at various times of the year, testing them to see when toxin levels are highest, whether it's during peak fish runs or when salmon are in short supply and orcas are burning fat, dumping the toxins into their circulatory systems.
And that's very, very important for understanding how to mitigate the problem, because it says that it may take a long time to clean up these toxins, but the most important thing to do in the interim is to keep the fish runs high and healthy so that they are less exposed to the toxins in their body.
While Tucker is out saving orcas, in a way, he's saving himself. The conservation canines mostly come from shelters. They're dogs who tend to be a little obsessive, especially about playing with balls. They sniff out scat and get rewarded with play. The scientists get data. Everybody wins.
It's really an unprecedented tool, because if you think about it, being able to get this information from multiple species at the same time, using our dogs, over such a very vast landscape at really frequent time intervals, is just incredibly powerful when you think about all the information that is being made available through feces.
That's it for this edition of UW360. You can catch this episode again on our website, uwtv.org/UW360, where you'll also find links to more information on everything we've covered today. Thank you for watching, and I'll see you next time with all news stories from the University of Washington.
Wardrobe for Carolyn Douglas is provided by C'est La Vie, located in downtown Edmonds and in Renton at The Landing.